Hi, I am Miss Prabhavati SN. Welcome to EPG Pathashala. In today's module, I would be explaining about vitamin A-like substances or the pseudovitamins. In this, you will be learning in detail about its properties and the functions. Vitamins are considered as one of the most essential organic micronutrients that have to be supplied through the diet. Apart from fat and water soluble vitamins, several other substances also exist in nature which have been included under the category of vitamins, although their definite vitamin character has not yet been clearly established. For some species, these vitamins may be regarded as most essential as an important metabolite, while some vitamins need not be required to be supplied through the diet because they may get synthesized within the body via tissue synthesis. For instance, vitamin C and Myo Inositol could be considered to fall into this category where it gets synthesized within the body. Only some species such as fish, gerbils and other types of species. During 1930s and 1940s, the compounds that were described as vitamins have been proven to be identical to other essential nutrients, or it can be a mixture of various compounds. In early days, the compounds which were referred as vitamins or growth factors or accessory factors are no longer called as vitamins. Certain compounds such as Myo Inositol, lipoic acid, coenzyme Q and polyphenols are known to possess biological activity, but they are not regarded as dietary essential for majority of the species. Basically, the term vitamin indicates that they are very essential for the maintenance of health and vitality. Pyrroloquinoline quinine is a newly discovered vitamin which needs to be subjected for thorough evaluation for it to be designated as vitamin. On the other hand, pangamate, lytril, gerovital and cabagin are generally not regarded as the dietary essential and are more appropriately called as pseudovitamins. After studying this module, you would be able to define the basic definition of pseudovitamins, define its physical properties and structure of various vitamin-like substances, understand the metabolic pathway, determine the effects of deficiency, understand the importance of other miscellaneous substances. Okay, now let us first study about choline. It is a dietary component needed for normal functioning of all cells. It is required for ensuring the structural integrity and signaling functions of the cell membranes. Prolonged ingestion of a diet devoid of choline is known to cause several consequences, including renal, hepatic, pancreatic, memory and growth disorders among mammals. Let us learn what are the major dietary sources. It is present in a wide variety of foods that are known to have membranes such as liver, eggs and wheat germs. It exists in both free as well as in esterified forms. Human milk is the richest source. Soy-derived infant formulas have lower total choline concentration. Metabolism. Choline is absorbed in small intestines. Free choline enters the portal circulation, which is mostly taken up by the liver, while the lipid-soluble phosphatidylcholine and sphingomyelin enters lymph and bypass the liver. Hence, it could be said that different forms of choline to have different degrees of bioavailability. After absorption, a small portion of choline is acetylated to acetylcholine by the enzyme acetyltransferase. Now let us understand the major functions of choline. It facilitates the synthesis of membrane phospholipids. During the early stages of pregnancy, choline is needed for supporting the normal neural tube closure. Any impairment in choline uptake and metabolism is proposed to be associated with the development of neural tube defects. Phosphatidylcholine and sphingomyelin are known to play an important role in signal transduction mechanism since they are known as the sources of lipid second messengers. Phosphatidylcholine is also utilized for the formation of lipoproteins. Deficiency. Deficient humans generally develop fatty liver due to the lack of phosphatidylcholine. Deficiency can also cause liver damage. Severe deficiency leads to the accumulation of homocysteine, which is known to increase the risk of cardiovascular diseases. Women who do not consume adequate amounts during pregnancy are known to give birth to a child with neural tube defect. Requirement. Studies have shown that premenopausal women have a lower risk for developing choline deficiency because they have the enhanced capacity for de novo biosynthesis of choline moiety. Information pertaining to its exact requirement is very scarce. Next, moving on to study the properties of Myo Inositol. It is also called as inositol, which is a water-soluble factor which do not possess any coenzyme function. Initially, it was isolated in muscle in 1850 and was defined as growth factor for yeast and molds. Deficiency in mice is characterized by inadequate growth, alopecia, and death. Evidences have proven that Myo Inositol is not a true vitamin for a large number of species. Nevertheless, the signs of deficiency have been demonstrated in certain species of fish and gerbils. Often it is very difficult to demonstrate the deficiency and is mainly associated with endogenous synthesis, variable turnover rates, and its interaction with certain vitamins and nutrients which could impair the demonstrable effect. Chemical structure and its properties. It basically exists in 9 forms and is a cyclohexane compound, out of which only Myo Inositol is known to exhibit biological activity. It is an alcoholic compound having hexose sugar. Pure substance is a white crystalline water-soluble compound having a sweet taste. It is stable to acids, alkalis at a temperature up to 250 degrees centigrade. Due to the presence of hydroxyl groups, it readily forms esters, ethers, and acetates. Myo Inositol in its hexaphosphoric acid ester form is reported to form complexes, particularly with phosphorus and other minerals, lending them unavailable during the process of absorption. And this compound is predominantly known as phytic acid. Now let us understand the metabolic system of Myo Inositol. In both humans and rats, the extent of absorption of ingested Myo Inositol is reported to be over 99%. The absorption is said to be less than 50% if the Myo Inositol is from phytate complex, especially in the presence of highest amount of calcium. The dietary Myo Inositol is mainly absorbed by active transport system. Studies conducted on animals have shown that it can be even converted to glucose. The metabolism of Myo Inositol is known to undergo 3 metabolic fates: the oxidation to carbon dioxide, utilization in gluconeogenesis, and it can also act as a facilitator for the synthesis of phospholipids. Now the functions. Its major functions are not so well defined. Certain biochemical functions could be probably related to its role in membranes and lipoproteins as a phospholipid component. It is a structural constituent of phosphoinositides and also acts as a membrane component. In addition to this general role, it is also been hypothesized to regulate certain cell surface phenomena. Most importantly, the binding of hormones and transfer of nerve impulses. This particular function is postulated to be taken care by phosphatidylinositol and its highly charged phosphorylated forms. Inositol 1,4,5-triphosphate has been recognized to play an integral role in the mobilization of cellular calcium via facilitating the release of intracellular calcium from the endoplasmic reticulum. Phosphoinositides have been reported to regulate several cellular processes including amylase secretion, insulin release, contraction of smooth muscle, liver glycogenolysis, platelet aggregation, histamine secretion, and synthesis of DNA in fibroblasts and lymphoblasts. The major food sources. It exists in measurable amounts in all plant and animal species. Foods that consist of seeds such as beans, grains, and nuts are considered as the most concentrated sources. Cantaloupe and citrus fruits are also reported to contain good amount of Myo Inositol. Plant sources generally contain it in a bound form to phosphate as phytate. Organ meats are one of the best animal sources in which Myo Inositol exists in a free form or may be present as a component of phospholipids. Extremely high concentrations may be found in heart, kidney, liver, spleen, and thyroid. Requirement. Humans and most species of animals that were studied have not yielded any information to draw conclusion with respect to the dietary requirements. This could be due to the fact that it is abundantly available and can also be synthesized endogenously, as well as bacteria is also capable of synthesizing. Carnitine, which is another pseudovitamin. It is essentially not considered as a vitamin because it can be synthesized in adequate amounts within the body tissues. Certain mammals have been demonstrated the need for providing supplemental carnitine only under certain circumstances in which biosynthesis is limited due to the nutritional deprivation of the major precursor amino acid such as lysine and methionine. It was isolated and identified in 1950 from meat extract, but its biological activity was determined during the 1960s. The basic structure and its properties. It is a quaternary amine. It is highly hygroscopic and easily dissolves in water. It exists in biological compounds as free carnitine as well as in ester form. It occurs in two forms as L and D carnitine, of which only L carnitine is considered as biologically active. Metabolism. Among omnivores, about 70 to 80% is reported to be absorbed from the diet under normal conditions. Absorption occurs across the proximal small intestine via active process as well as passive diffusion mechanisms. The uptake from the intestinal lumen into the mucosa is very rapid. Out of the total amount, about half the amount undergoes acetylation in the tissues. Carnitine is not circulated in blood. The cardiac and skeletal tissues require carnitine for the normal metabolism of fuel. The uptake from the heart is facilitated by cardiac carnitine binding protein. Liver and kidney are the major sites for the synthesis of carnitine. Skeletal muscle serves as the major storage site. The major route of excretion is through urine. Human kidneys conserve and reabsorb more than 90% of filtered carnitine and thus is considered to play an important role in the regulation of carnitine concentration in the blood. Moving on to study the major functions. It acts as an important cofactor for normal cellular metabolism. Adequate carnitine stores are known to promote the optimal utilization of fuel substrates. It is also needed for the transport of long-chain fatty acids into the mitochondrial matrix. It protects the cells from the accumulation of toxic acyl-CoA compounds either from endogenous or from exogenous sources. It is been evaluated to exert protection on human cells from the effects induced by oxygen-free radical damage. It plays an integral role in nitrogen metabolism. It also acts as an ammonia detoxifier and thus protects the ischemic heart from reperfusion injury by scavenging free radicals. It is also considered as a memory and alertness boosting agent in patients suffering from Alzheimer's disease. Requirement: certain higher animals as well as mammals can synthesize carnitine. Humans fed on low carnitine diets would contain low tissue stores. This may impair the biosynthetic process. Normal adults can ingest carnitine to the extent of 100 mg per day. Healthy children and adults can synthesize 20 mg of endogenous carnitine on a daily basis. The major precursor for carnitine synthesis in mammals is gamma-butyrobetane, which can be synthesized from most tissues with the help of the amino acids like lysine and methionine. Food sources: the foods of plant are generally known to contain lower amounts. Animal-derived foods are one of the richest sources. Dairy products also contain considerably higher amounts. The carnitine concentration could be represented in an increasing order as fish, poultry, pork and beef. The darker the meat, higher is the concentration of carnitine. Typical concentration in beef meat could be 600 micrograms per kg, 40 to 90 micrograms per kg in chicken and 75 microgram per kg in lamb respectively. The next pseudovitamin is Coenzyme Q10. In the year 1955, a group of scientists isolated an unsaponifiable lipid which was shown to have ultraviolet absorption property in the intestinal mucosa of horses. This new substance was identified as quinine, which was found to be widely distributed in animal tissues and thus was named as ubiquinone. 2 years later, scientists from University of Wisconsin isolated a novel quinine in the lipid extracts of mitochondria, which was later named as coenzyme Q because it takes part in the electron transport chain. Now let us study the properties. It is chemically known as 2,3-dimethyl 5-methyl-6 polyisoprene parabenzoquinine. It occurs naturally as an orange lipophile powder which is odorless and tasteless. It has 10 isoprenoid units and thus is called as coenzyme Q10. It is not a very stable compound and degradation may occur at a temperature of 46 degrees centigrade. Q10 form is most abundantly found in humans, animals, microorganisms and plants. It primarily exists either as cis or trans form. Trans form is proposed to be widely found in nature. The major food sources: it exists in a wide variety of foods in small amounts. Among animal foods, pork heart is found to contain 126.8 micrograms per gram, followed by slightly lesser amount in beef heart having 113.3 micrograms per gram. The amount in chicken meat is very negligible. Traces may be found in egg. Vegetables such as peas, cauliflower, beans and carrots contain 2.7, 1.8 and 1.7 micrograms per gram respectively and are considered as the poor sources. Fruits and dairy products are also known to contain very negligible amounts. Q10 levels in human tissues. Concentration is highly dependent on age, sex, race and the health of an individual. Plasma or serum Q10 is generally employed as a means of assessing Q10 status in humans. The blood values among healthy volunteers is known to be in the range of 0.4 to 0.45 micrograms per ml. Among Asian males, the plasma concentration is reported to be significantly lower. Hence, this could be considered as one of the major risk factors for increased incidences of coronary artery diseases among Asian Indians. It also plays a major role as an antioxidant. In its reduced form, that is as hydroquinone, it functions as a lipophilic antioxidant and has assumed greater importance due to its free radical scavenging activity. It promotes the stability of cell membrane as well as prevents DNA damage induced by free radicals. It has the greatest potential to recycle and regenerate other antioxidant substances, mainly tocopherol and ascorbate. Pharmacological benefits. It could be used to treat several cardiovascular conditions, can be used to treat hypertension. It acts as an anti-cancer as well as immune stimulating agent. Q10 supplement is beneficial for correcting periodontal and respiratory diseases. Supplementation is also known to improve glycemic control. It helps to slow down the progression of Parkinson's disease in the early stages. The progression of rheumatoid arthritis could be slowed down with Coenzyme Q10 supplements. Let us study about the major uses of other additional vitamin-like substances. First one is para-amino-benzoic acid. It was originally identified as growth factors for many species of bacteria. It is an integral part of folacin molecule. It exerts a unique property of counteracting the bacteriostatic effects of sulfonamides. It is used as a protective component in sunscreen lotions. Lipoic acid. It has gained particular importance as an agent required for promoting growth of certain microorganisms. It is required for the oxidative decarboxylation of alpha keto acids like pyruvic acid during the metabolism of carbohydrate. So far, there are no clear evidences available about the established need in animal nutrition. Vitamin B13 or orotic acid. It was first isolated from distillers soluble and the purified compound was called as orotic acid, which acts as an intermediary compound in pyrimidine metabolism. It has no known coenzyme function. Vitamin B15 or pangamic acid. It is present in rice bran and yeast. There are no established evidences to confirm its role as having vitamin activity or to afford therapeutic benefit. It is basically not a chemically defined substance. Vitamin B17 or amygdalin. It is a beta-cyanogenic glucoside, occurs naturally in the kernels or seeds of almost all fruits. It is known to possess a specific role, that is, it helps to destroy cancer cells. The presence of cyanide in laetrile is thought to be responsible for this particular property. Vitamin H3 or gerovital. It is a buffered solution of procaine hydrochloride. It is also known as novocaine, a painkiller used by dentists. It also helps in alleviating the symptoms of age-related diseases. Thus, it is being promoted as a nutritional substance. But yet, there are no clear scientific studies in this regard, and hence it was declared that it cannot be called as a vitamin. Vitamin U or carbagen. It is known as an anti-ulcer factor. It is found naturally in cabbage and other green vegetables. Methyl sulfonium salt of methionine is the active substance which functions as an anti-ulcer factor. However, the claims have not been supported through the scientific studies. Okay, now let us summarize today's episode. Vitamins are considered as one of the most essential organic micronutrients that have to be supplied through the diet. Certain compounds such as Myo Inositol, lipoic acid, coenzyme Q and polyphenols are known to possess biological activity, but they are not regarded as dietary essential for majority of the species. Inositol, which is a water-soluble factor which do not possess any coenzyme function. It exists in measurable amounts in all plant and animal species. Carnitine is essentially not considered as a vitamin because it can be synthesized in adequate amounts within the body tissues. Coenzyme Q10 was identified as quinine, which was found to be widely distributed in animal tissues and thus was named as ubiquinone. Other miscellaneous substances are also known to possess vital bioactivities. Thank you students. Hope you have understood today's episode.